Hello and welcome. Today let's talk about our special senses. We'll go through our sensory physiology. And before we do that, we'll hit at the anatomy and get the anatomy taken care of so that way you understand exactly where we are at, where we're going, and what's happening in relation to the signals. Now, when we talk about our special senses, we'll be looking at our special sensory receptors there then in greater detail as well. Now, they're going to be distinct, localized receptor cells found in the head, found in relation to our head. Now, the special senses include, as we can see here, vision, number one, taste, we have smell, sense of hearing, and equilibrium, So what we'll do is we'll start off with the eye and we'll go through vision and then we'll get vision taken care of and then we'll move through and we'll go forward with the rest of the senses. So here then we can see when we talk about the eye and vision, first thing I'd like you to know is that 70% of the body's sensory receptors are going to be found located in the eye. And these receptors we're going to look at in greater detail and we'll see are going to include your rods and your cones. Your rods and your cones. We'll see most of the eye is going to be protected by a cushion of fat and then the bony orbit that you've already studied, I'm sure, in anatomy. So here, let's go through and we can see we've got basically the eye. Here you can appreciate a few structures in relation to the eye from just an external view. And you can see when we look at this picture of the eye, really all that's visible is basically a very small percentage of the eye. The rest of it is all hidden. That's kind of like the respiratory system. The only part that's visible is this external nose. The rest of it is all hidden. So here with the eye, you can see the majority of it is all hidden and we can see a small percentage. Now here we can see first the accessory structures. We've got the palpebrae, the superior and the inferior eyelids. And then these eyelids, you've got these eyelashes projecting from their free margin. And these eyelashes are there to protect the eye. And then here you can identify a few structures. So here then we can appreciate the lacrimal caruncle. And from your anatomy knowledge, you should remember this is found at the medial commissure. So here this is telling you that the nose should be right inside of here. This is the medial aspect of the eye. And this is the lateral aspect of the eye. Next then you can appreciate here we have our iris, our pupil. And then we've got our conjunctiva basically here on the sides. Let's go through and let's look at the eye in greater detail now. When we talk about the eye, first what we'll do is we'll go through and we'll look at the different layers of the eye. So now when we go through and we look at the structure of the eyeball, we'll see the eyeball is a hollow sphere that's going to be filled with fluids that are called humors. The eye is going to be divided up into three layers, and those three layers will include the fibrous layer we can see here first, vascular layer then number two, and third then we have our inner layer. The lens is going to separate the internal cavity into the anterior and the posterior segments, basically into cavities. So here's the lens. We said it separates, right, the internal cavity into an anterior and a posterior segment. So anterior is going to be found in front of, and then posterior segment behind, right? Just the terms anterior and posterior give that away. And here we can see the posterior segment is going to be filled with a vitreous humor. It's a gel-like material I'm sure you learned about in anatomy. Vitreous humor. And then we're going to have, we'll see aqueous humor. We'll have aqueous humor in the anterior segment. Aqueous humor. Next in here, let's go through and let's talk about first the fibrous layer. So here coming back, you can see the first layer is right outside here, the fibrous layer. Makes its way all the way around. Next, then we're able to appreciate the vascular layer. The second layer is the vascular layer. So here you can see it's been cut, so you can appreciate that vascular layer there, and you can see how it makes its way all the way around also. And then third, we can appreciate the inner layer. The inner layer, and the same thing with the inner layer, it's been cut, and you can see it making its way all the way around. That is the inner layer. Let's talk first about our fibrous layer. Let's talk first about our fibrous layer. Now, when we talk about our fibrous layer, the fibrous layers we can appreciate is the outermost coat of the eyeball And it consists of two regions. The first region we can appreciate is this region right back here, the sclera. And then second, we can appreciate the cornea. The cornea. Let me turn the... So here we can appreciate the sclera, and then anterior to the sclera, we're going to have the cornea. The cornea. So again, it's the outermost layer, this fibrous layer is the outermost coat. It's the outermost coat of the eyeball, we said, and it consists of two regions. You've got the sclera, all back here, and then anterior to that you can appreciate the cornea, the two divisions. It's composed of, we'll see this fibrous layer is composed of dense avascular connective tissue. It is composed of dense avascular connective tissue. Let's talk then about the sclera in greater detail. Now the sclera, you can see it forms the posterior portion or the posterior bulk of the fibrous layer. It functions to protect and shape the eyeball. It also provides an anchoring site for the extrinsic eye muscles. And here we can see it's pierced posteriorly by the optic nerve. It's going to pierce it as it makes its way through the eye. So next we can appreciate the cornea. The cornea makes up the anterior sixth of this fibrous layer. It makes up the anterior sixth of this fibrous layer. So it's not occupying too much space there. It is crystal clear and it forms a window that's going to allow light to enter into the eye. And we'll see it's a major part of the light bending apparatus of the eye. We'll check that out when we look at the physiology. Next, then let's move into the vascular layer. So when we talk vascular layer, the vascular layer now we'll see forms the middle coat of the eyeball. It forms the middle coat of the eyeball, and it consists of three regions. First, we've got the choroid. We can appreciate right inside of here, the choroid. Anterior to the choroid, then we're going to be able to appreciate the ciliary body. And then anterior to the ciliary body, we're going to have the iris. So the three divisions then of the vascular layer. Let's talk first about the choroid. Now when we talk about the choroid is a blood vessel rich membrane, and it forms the posterior majority of the middle layer, the vascular layer. Its blood vessels are going to nourish all the layers of the eye. It also has brown pigment, and brown pigment is a chemical that has the ability to absorb light. And this brown pigment came from melanocytes. It's going to help absorb light and prevent it from scattering within the eye, which would lead to visual confusion. So very important there. Next, then we've got our ciliary body. So next, then moving forward, we've got our ciliary body. When we talk about the ciliary body is a thickened ring of tissue that's going to be found encircling the lens. It consists of smooth muscle bundles that are going to control the shape of the lens. Moving anterior to that is the iris. Now when we talk about the iris is the visible colored part of the eye. It forms the most anterior portion of the vascular layer. It contains a round central opening. You can see right inside of here. That's called the pupil. That's called the pupil. You can see that right inside of here. And this pupil is going to allow light to enter the eye. The iris, we'll see, is made up of two smooth muscle layers. In close vision and bright light, so you can see here, we've got our two sets of smooth muscle, our sphincter pupillae and our dilator pupillae. So we can see when the sphincter pupillae muscles contract, this is going to decrease the pupil size. And when the dilator pupillae muscles contract, this increases the pupil size. So again, as I was saying, in close vision and in bright light, the sphincter pupillae, the circular muscles, they're also known as, contract. And when they contract, the pupil is going to constrict. In distant vision and dim light, the dilator pupillae, the radial muscles, contract and the pupil dilates, allowing more light to enter the eye. And you can see that there very nicely. And these are also examples of parasympathetic and sympathetic response. So next then let's move to the inner layer. Now when we talk about the inner layer, we're talking about the retina. So let's move back to this picture right inside of here. So here you can appreciate that retina now. This retina is going to help form the innermost layer of the eyeball. This retina is very delicate. It contains millions of photoreceptors that convert light energy. The retina consists of two layers. Let's go through and let's zoom in on that retina now in greater detail. So we'll move over to this picture and you can see the two layers. First we have our neural layer, and then second we have our pigmented layer. You can see the pigmented layer is in between that neural layer and the choroid. So it goes sclera, choroid, pigmented layer of the retina, and then the neural layer of the retina. Now when we talk about the pigmented layer, we'll zoom in on this even further. So here you can see now only the retina. Here we have that pigmented layer, and then you can see after that is all neural layer. Neural layer. All neural layer. Now when we talk about this pigmented layer is the outer single cell thick lining. The pigmented layer, they say it abuts the choroid. It's found right next to the choroid, and it extends all the way to the back of the iris. And it extends all the way to the back of the iris. So here you can see where that iris is at. All the way to the back of the iris. These pigmented cells, they're like the choroid, and they absorb light and they prevent it from scattering in the eye, again, which would lead to visual confusion. These cells also act as phagocytes, and they store vitamin A. Next, then, let's look at the neural layer of the retina. Here we can appreciate first our photoreceptors. Second, then, are our bipolar neurons, our bipolar cells. And then third, we've got our ganglion cells. And third, we have our ganglion cells. Signals get produced in response to light. And they're going to spread from these photoreceptors to the bipolar cells and then to the innermost ganglion cells where action potentials are generated. The axons we can see here of the ganglion cells eventually make these right hand turns, these right angle turns, and they leave the posterior aspect of the eye as the optic nerve. So you can see that here also. Here's the ganglion cells, and you can see their axons are all making right angle turns, and they're leaving this posterior aspect of the eye as the optic nerve. When we talk about a nerve we've said is a bundle of axons. So that's what you're seeing here. Then we have the optic disc you can see here. This is where the nerve exits the eye. This is the weak spot on the fundus, on the posterior wall of the eye, the fundus. This is a weak spot because it is not reinforced by sclera. The optic disc is also known as the blind spot. And you can see here why, because there are no photoreceptors in this area. You can see the photoreceptors stop about here and right about here. The rest of that space is occupied by this optic nerve. So there's no photoreceptors here, so we refer to this region as the blind spot. The optic disc is the blind spot. So the optic disc we can see is the blind spot. You might say, wait, hold on, I can see everything. I'm not, blind in any region. It's because our brain is going to fill in all the gaps from memory. Our billions of photoreceptors are going to be found in the neural layer, and they are of two types. First, we have rods, we said, and second, we have cones. Now, when we talk about our rods will perceive gray tones, gray colors. They are our dim light and peripheral vision receptors. Our rods are. They are our dim light and peripheral vision receptors. So we can see here, they are more numerous in quantity compared to the cones, and they are more sensitive to light compared to the cones. So next in here we can appreciate our cones. And when we talk about our cones are going to be vision receptors that require bright light. So they're going to get activated in bright light, and they provide us with high resolution color vision. They provide us with high resolution color vision. We have three different types. We have red cones, blue cones, and green cones. And we have an overlap of these cones and how our brain integrates these incoming signals. This allows us then to see lots of different colors. Now we can see lateral to the blind spot then, we're going to be able to appreciate the macula lutea. So lateral to the blind spot, we've got the macula lutea. The macula lutea has a minute pit at its center, and the macula lutea has a minute pit at its center, and this pit is called the fovea centralis. The fovea centralis has a high concentration of cones. The fovea centralis has a high concentration of cones, which provides us with detailed color vision. Next, let's talk about the lens. The lens is a biconvex, meaning curved, transparent, flexible structure that can change shape to precisely focus light on the retina. And we'll see the lens has two regions, the lens epithelium that's found located anteriorly, and the lens fibers, which are going to form the bulk of the lens. Then we can see the lens fibers are filled with transparent protein called crystalline. The lens becomes more dense, convex, and less elastic with age. And here's one condition that can occur in relation to the lens, and it's called cataracts, basically a clouding of the lens. And it usually happens due to aging, diabetes mellitus, heavy smoking, and frequent exposure to intense sunlight. And here we can appreciate a picture of a cataract, the clouding of the lens. Right? If we have clouding of the lens we described first, let's go back here. The lens I described to you as being transparent. So you have clouding. Let's come to this picture here. So the lens, so here we can see the lens. The lens is supposed to be transparent, but if you have clouding in there, how is light going to be able to pass and make its way to those photoreceptors that are way back here? So that's why it becomes problematic. All right, next here then let's talk about optics and light in relation to the eye. Now, when we talk light and optics, in order for you to understand the function of the eye as a photoreceptor organ, we need to understand the properties of light first. Now, when we talk about our eyes are going to respond to visible light, which is going to be a small portion of our electromagnetic spectrum. And visible light's going to have wavelengths of about 400 to 700 nanometers. First, let's understand electromagnetic radiation. Here we can see this chart. This chart includes all energy waves from long radio waves all the way to very short gamma waves and X-rays. Examples include gamma radiation, right? X-ray radiation we use for X-rays. UV radiation, right? We need for vitamin D conversion, but then too much causes burns, as we've seen. Visible radiation, that's visible light, that's what our eyes are going to respond to. And then we have infrared radiation, microwave radiation, obviously we know, and then radio waves. Our eyes respond to a part of this spectrum that's called visible light. Visible light travels in the form of waves, and its wavelengths can be measured very accurately. As we can see here, scientists describe light as packets of energy, photons, as packets of energy. So scientists describe light as packets of energy, photons, that are traveling in a wave-like fashion at very high speeds. When visible light or sunlight passes through a prism, its component waves bend to a degree, dispersing the beam of light into a visible spectrum or a band of colors, which then we're able to see. And here we can see those colors. Those colors include red, orange, yellow, green, blue, indigo, and violet. There's a mnemonic for that, right? Roy G. Biv. Red, orange, yellow, and then going all the way to both. Like sound, light can reflect or bounce off of a surface. This reflection or bouncing of light from or off objects in our environment accounts for most of the light reaching our eyes. Objects have color because they absorb some wavelengths of light and reflect others. A red apple, for example, is going to reflect mostly red light, while green grass reflects green. Things that look white reflect all wavelengths of light, and black objects absorb them all. So white doesn't absorb any colors. Black absorbs all lights. You can see a red shirt because it absorbs all colors except red. Red is going to bounce off, and we see red. Next, then, let's talk about refraction and lenses. Now, here I would like you guys to know that when light travels, it can be blocked by a non-transparent object. When light travels in a given medium, its speed is constant. Okay, when light travels in a given medium, its speed is constant. But when light passes from one transparent medium into another with a different density, its speed changes. Light speeds up as it passes into a less dense medium, and it slows as it passes into a denser medium. Because of these changes in speed, bending or refraction of a light occurs when it meets the surface of a different medium at an oblique angle rather than at a right angle, which would be perpendicular. So here in this figure, we can see refraction occurring. A straw in a glass of water appears to break at the air-water interface. When we spoke about the lens, we said that the lens is a transparent object and it is curved. When light rays pass through the lens, they hit the curves at an angle and bend. You have refraction of the light rays occur. They're going to then converge. They are then going to converge or intersect at a single point known as the focal point. The focal point is a point at which light rays meet after passing through a convex lens. The image formed by a convex lens is called the real image, and it's inverted, meaning it's upside down and reversed from the left to the right. Our brain then has the job of flipping the image. Let's move forward then. Let's talk about focusing light on the retina. When light passes from the air into our eyes, it moves through the cornea, the aqueous humor, our lens, our vitreous humor, and then through the neural layer of the retina. Next, it makes its way to the pigmented layer to excite our photoreceptors. During light's passage, light is going to be bent 3 times. The first time it gets bent is when it's going to be entering the cornea. The second time is when it's entering the lens. And the third time, leaving the lens. The lens is a highly elastic structure, and its curvature and light bending power can change to allow fine focusing. So now when we talk about focusing for distant vision, our eyes are best adapted for distant vision. To look at distant objects, we only need to aim our eyeballs so that they are both fixated on the same spot. Next, we can talk about the far point of vision. I'd like you to know our far point of vision is the distance beyond which no change in lens shape is needed for focusing. For the normal or emmetropic eye, we can refer to it as, the far point is 20 feet. The far point is 20 feet. It means basically anything after 20 feet doesn't require the lens to change shape. And here we'll see the ciliary muscles are going to be relaxed, causing the lens to be stretched flat by the tension in the ciliary zonules. So here you can see that. The lens is flattened for distant vision. This also is mimicking a sympathetic reflex activation. So here we can see sympathetic input relaxes the ciliary muscles, tightening the ciliary zonule and flattening the lens. And then when we talk about focusing for close vision, you'll see light from close objects diverges as it approaches the eye and it comes to a focal point farther from the lens. For this reason, close vision demands that the eye make active adjustments. So in order to restore focus for close vision, we'll see three processes must occur simultaneously. Number one, accommodation of the lenses has to occur. This is a process that causes the lens to bulge and produce a shorter focal length needed to focus the image on the retina. Number two, then, we have a constriction take place. We have a constriction of the pupils. Number three, we have convergence of the eyeballs occur, where we have medial rotation of the eyeballs by the medial rectus muscles. So here let's look at the three adjustments then in greater detail as we spoke about. So accommodation, we said, is a changing of the lens shape to increase refraction. So here I'd like you to also know about the near point of vision. The near point of vision is the closest point on which we can focus clearly. And it represents the maximum bulge the lens can deliver. And it represents the maximum bulge that the lens can deliver. In young adults, it's usually 4 inches from the eye. Now, when the lens becomes non-accommodating, usually after the age of about 50, it's called presbyopia or old person's vision, where you'll see they're having to use glasses for objects that are up close. Next, then, we have constriction we talked about. And third, convergence. So here, let's look at that. Again, it mimics a parasympathetic reflex activation. Parasympathetic input contracts the ciliary muscles, loosening the ciliary zonule, allowing the lens to bulge compared to what we had in distant vision.